Hi, my name is Taylor Vidopia, and I'm a master's student at the University of Waterloo. And today I'm going to be talking about nutrient cycling on restored seismic lines in peatland. So in Western Canada, we have a lot of tree peatlands, and a lot of these, especially in Alberta, are disturbed by seismic lines. What is a seismic line? Well, seismic lines are created for oil and gas exploration. What happens is they come in and they remove the vegetation so they can get their seismic equipment onto the landscape. This sends vibrations below the surface, and depending on how they are absorbed and reflected, can tell them whether or not there's natural resources beneath the ground. As you can see in this picture, these little white squiggly lines are seismic lines. They really fragment the landscape. Seismic lines began to be created in the early 1950s, and still to this day, they're really disruptive and they don't really, vegetation basically has a very hard time growing back on these lines, especially trees. So why are plants having such a hard time growing back? Could it be because of a lack of nutrients? Well, we all know that plants like us need nutrients to grow. So my objective in my study is going to be to assess the impacts of different seismic line restoration treatments on nitrogen and phosphorus, the bioavailability, as well as mineralization rates in a peatland directly after restoration, which I did last summer, and 1 year post-restoration, which I hopefully will get out to do this summer. My site is on a peatland, and it's located in the Alberta Boreal Forest. It has 2 intersecting seismic lines, and we use 2 different kinds of restoration treatments on it. We used ripping as well as mounding techniques. It's really important to restore the site to get the natural topography back. We want these bumps or lumps on the landscape called hummocks, and these dips or depressions known as hollows. This is really important because when a seismic line is created, the peat is compacted, causing the water table to increase and the peat to become really saturated, which makes it hard for woody vegetation like trees that don't really like to be submerged to reestablish on the line. So introducing this microtopography or microforms of hummocks increases the aeration, decomposition, and really gets that microbial activity going again, which increases the biodiversity. It's really important still to have those hollows because we want that water-loving vegetation like sphagnum to still thrive. So that's why we did these 3 different restoration techniques. The first one was a rip and lift. So it's just how it sounds. We're ripping the peat back to form that hummock, and the ripped area is acting as a hollow. Then we have 2 kinds of mounding. First, we have inline mounding, where we excavate or take a bucket of peat from the line and place it directly beside that, with the vegetation side up. And then the second type of mounding, we take a hummock from off the line in the natural area and place it onto the line, with the vegetation side up, hopefully to get the vegetation to grow and establish quicker and restore that line faster. We also compared this against an untreated part of the line where there was no restoration done, and a natural off the line where there was no disturbance. We collected peat samples as well as pore water samples to look at ammonium, nitrate, and soluble reactive phosphorus mineralization rates and concentrations. Our initial results are showing that there actually is no significant difference between the treated, untreated, and natural area for the nutrient analysis, except for we did see slight significance between the months for ammonia mineralization rates. So really, there was no significant difference between the treatments, untreated, and natural sites observed for the nutrient analysis. But this is really important because these results gave us background concentrations for the nitrogen and phosphorus pools and mineralization rates, so that as this peatland recovers, we can see the progress of these restoration techniques, so we know how to best restore seismic lines. Thank you.
Hi, I am Tasha Rabinowitz. I'm in Halifax, in Nova Scotia, and I'm just going to talk quickly about my master's research on salt marsh restoration. So salt marshes, if you were here yesterday for the student talks, we learned a little bit about them, but basically, just as a refresher, they're a type of tidal wetland, and so this is just a picture of one. You can see this is a tidal river, and so that tidal hydrology is really important to those systems, and this is at high tide, so they'll all be flooded at high tide. And so any sort of restrictions to that tidal hydrology can result in a pretty major loss or degradation of these types of ecosystems, and this is just kind of an example of a tidal restriction. You can see this is a bridge. It's obviously way too small for this crossing, where this marsh back here should be flooded, more flooded than it is. And in Atlantic Canada, where I do my research, it's estimated that up to like 90% of the historic range of these salt marshes has been lost, which is a pretty incredible amount of loss. And so when we're talking about restoration, usually the first thing that you want to do is restore the hydrology. Obviously, that's the most important thing. So to take out the restriction like that. And often the projects that I'm working on here, usually they'll just kind of let the natural processes drive the recovery on its own. And frequently that works. But often also what we'll see is a longstanding deficit in vegetation diversity. So the plants that are coming back, we're not necessarily seeing the communities that we would want to see at a natural healthy marsh. And so this is a picture of one of our restoration sites. This is just the first year, early in the year, but this is kind of what they look like right after they get restored, which is interesting. And so, yeah, so I guess the solution to this problem of this deficit in vegetation diversity is to plant plants. And that's kind of where my research comes in. That active restoration of vegetation is frequently a part of restoration projects, but we haven't really done it here in the Bay of Fundy area. And so just to situate you as to where I am, I'm in Nova Scotia here, and then around the Bay of Fundy, I'm working on 2 sites where the tide has been restored within a year of starting this project, so 2 years ago, I guess. And the Bay of Fundy has some sort of unique characteristics that make it important to test these sort of revegetation techniques here. And so my project, what I did at these 2 sites was to basically just add plants in a bunch of different ways. So on the left here, I established these sort of small plots and then added plants in a different way in each of them. And the treatments that I did were planting potted seedlings on the right, you can see, and in the middle, field transplants, so just digging up plants from the field and transplanting those. And then I also added seed in a couple different ways, so I just sowed seed directly. And I also planted this material called rack, which is basically just like a mat of dead vegetation. And it's naturally found in the marsh, and it's been shown to contain seeds in the past, so we just kind of put some of that in the ground to see if anything would come up. And then I kind of monitored the health and the growth of those plants. Cool. Over the course of the growing season. So this is from the end of last season, so the first growing season, and you can see that these sort of the potted seedling and the field transplants, so adding mature plants was really ended up with the most abundant, this is like cover of the target species by the end of the season, and that's no real surprise. If you add bigger plants, there will be bigger plants. But I didn't see a whole lot of germination from any of the seeds or any of those other like random treatments. And definitely no more cover than the passive sort of control treatment. And so taking a closer look at some of those, the mature plant treatments, the results were kind of interesting, and this is a lot to be on this slide, but what I really want to highlight is that survival, so just whether or not a plant survived to the end of the season, how they got added was really important. So those potted seedlings really survived a lot better than the field transplants. But looking at growth and health of those plants, and which are just sort of other plant performance measures, what we saw with that was that the growth and the health were, the site conditions were way more important than how we added them. And so any of those plants that really survived kind of had an equal chance of growing to be large and being healthy at the end of the season. And so with this research, I'm just kind of hoping to inform restoration practitioners, both in Nova Scotia and, you know, down the east coast of the States too. There are similar marshes. So just hoping to help to inform sort of best practices for restoration moving forward. And that is me. Thank you.
So, hi everyone, my name is Emily, and today I'm going to be presenting to you on my research studying the effect of sodium concentration on the rhizodeposition of Juncus balticus and Carex equatalis, which are two common peatland species. Before I go further, I just wanted to acknowledge that me being in Waterloo, I am on the traditional territory of the Neutral, Anishinaabe, and Haudenosaunee people, and I encourage everyone to consider what Indigenous lands they reside on and do research on, as well as what that means for actions that they can be undertaking. So many peatlands are affected by sodium or are expected to have sodium become an increasingly important factor. These include naturally saline peatlands, coastal peatlands that may be affected by sea level rise, as well as constructed peatlands in a post-mine landscape. Sodium can affect carbon dynamics through its influence on biota, as well as through its influence on chemistry. One way that sodium can affect plants is by influencing rhizodeposition. Rhizodeposition is the release of all organic carbon from plant roots, but for my research, I focused on the dissolved fraction. Once these compounds enter the rhizosphere, they contribute to dissolved organic carbon, can be decomposed and contribute to greenhouse gas emissions, or potentially be sequestered. My objective was thus to quantify and characterize the rhizodeposits of Carex equatalis and Juncus balticus under different sodium concentrations. To do this, I took plant cores from peatland and isolated the individual plants and grew them hydroponically under different sodium concentrations. I then took the hydroponic solution they growed in and analyzed it using liquid chromatography organic carbon detection. This method fractionates the carbon based on size, with the larger compounds eluting before smaller compounds. The compounds I'll be talking about today are biopolymers, which are polysaccharides and proteins, humic substances and building blocks, which for plants would be a variety of aromatic and polyphenolic compounds, and low molecular weight acids, which are organic acids. So these are my results. We have Carex equatalis on the left and Juncus balticus on the right. The y-axis shows the rhizodeposition rate, and then the x-axis has those different carbon fractions that I mentioned earlier. As you can see, both species have fairly similar rhizodeposition rates and have a very large contribution of those aromatic compounds and those biopolymers. Sodium concentration is indicated by the color, with red being the lowest sodium concentration and purple being the highest. What I found was that as sodium concentration increased, the amount of aromatic compounds tended to increase. Aromatic compounds can go into the plasma membrane and stabilize it, reducing diffusion, similar to how cholesterol acts. This would prevent sodium from passively diffusing across the membrane into the plant and prevent toxic accumulation. I also found a decrease in low molecular weight acids, or organic acids, and I suspect this to be due to the stabilization of the membrane from those aromatic compounds, as these compounds can passively diffuse across the membrane into the rhizosphere. So if we have a more stable membrane, then they won't be released at a higher concentration. Finally, I found a decrease in the amount of biopolymers, which are those larger compounds, but I suspect this might not be due to a direct effect of the rhizodeposition rate, but instead, once these large compounds, generally negatively charged compounds are released into the hydroponic solution, the high sodium concentration reduces charge density and allows them to precipitate, so they're not measured. Though this isn't a direct effect of rhizodeposition, it still indicates how these compounds will act once they enter the rhizosphere. So overall, I found a large contribution of biopolymers and aromatic compounds to rhizodeposits. I found evidence of plant physiological response to limit sodium uptake with those aromatic compounds, and I found that elevated sodium shifts rhizodeposition to a higher proportion of aromatic compounds as a contributor to dissolved organic carbon. If anyone has any questions, please don't hesitate to contact me and we can set up a meeting, or you can address me on the Slack channel. And before I go, I just wanted to acknowledge my supervisors, Dr. Maria Strack and Dr. Jonathan Price, as well as the people who helped me with fieldwork and the funders of this research. Thank you.
So hello again, my name is Miranda and the title of my talk is Environmental Drivers Influence Carbon Emissions from Vacuum Extracted peatlands. So what is peat extraction? Well, companies start with a pristine peatland like the one shown in image 1. They remove the surface vegetation, they install drainage ditches to lower the water table, and then they begin to extract this peat. In Canada, the primary method is vacuum extraction, where they vacuum suck up the top layer of the peat, they dry it, and then sell it. In Canada and the US, it's primarily sold for horticulture purposes. In Europe, it's also used as a fuel source. And a typical peatland will be extracted anywhere from about 20 to 35 years before restoration can begin. So why do we want to study carbon emissions during this active extraction period? Well, pristine peatlands are a net sink of carbon. And when we extract them, we turn them into a net source of carbon. And research shows that they release both carbon dioxide and methane emissions. Environmental drivers can help us understand the location and magnitude of these carbon emissions. Soil temperature is an important driver, and generally there's a positive relationship between them. Soil moisture is another important driver, and there's often a unimodal relationship between them. My work is part of a larger group of researchers at several universities across Canada, and my part of it is to understand the carbon dioxide and methane emissions from actively extracted peatlands in Alberta, Canada. And the objective I'm going to present on today is to assess the influence of environmental drivers on these emissions. Before I get to my sites, I want to acknowledge that my project takes place within Treaty 6 territory in Métis Nation of Alberta, Region 4. So this past summer, I did fieldwork in Alberta, and I've shown satellite images of my 3 sites, each of different age since extraction began. And you can clearly see the drainage ditches and just the level of destruction to this environment. I created 21 transects, which are shown in the yellow dots. And I positioned these transects perpendicular to the drainage ditches and took measurements 2 meters, 5 meters, and 15 meters from the ditch to try and create a hydrologic gradient, because we'd expect better drainage closer to the ditch and poorer drainage further away from the ditches. To measure these carbon emissions, I used the closed chamber method, and I also recorded those environmental variables, soil temperature and soil moisture. Some of my results, I found there was a positive relationship between carbon emissions and soil temperature. Here I've plotted carbon dioxide, but this was the same for methane as well. And this is in agreement with the general literature. I found that there was a positive relationship between carbon emissions and soil moisture at the young site, but not at the intermediate or old site. And the age there refers to how long extraction has been going on. The positive relationship suggests that we might be on the first side of that unimodal relationship, and so there's a moisture stress for the microbes. You can see that at the old and intermediate site, there's a very narrow range of soil moisture values, and I only recorded soil moisture for about 5 weeks over the summer, and so I wonder if, next summer when I can record it for the whole summer, if I'll see the same result or if I might start to see a positive relationship there. And finally, I was looking at the carbon emissions along my transect, where I was measuring 2, 5, and 15 meters from those drainage ditches. And in this figure, you can see that there was no significant difference in these carbon emissions with distance from the ditch. The distances are shown with the different colors, 2, 5, and 15 meters. One reason for this is that it was a very wet summer, and so I also didn't see those hydrologic gradient. There was no difference really in soil moisture or water table along that transect. So I wonder in a more normal year if we start to see the difference in drainage and thus maybe see differences in these carbon emissions. So I want to thank all my funding sources, my supervisor, everyone who helped out. And yeah, if you have any questions, feel free to contact me. Thanks.
greetings from Macedonia, a little bit over the world. So thank you all for the great presentations and the great sessions before. I really appreciate it. Before I start my presentation, and I would just quickly like to roll you over the globe. So as you can probably see on, oops, I don't know if you, okay. So as you can probably see on this Google Earth image, I'm just trying to show you where Macedonia is. You probably know where Greece is here in Europe. So Macedonia is this small country at the center of the Balkan Peninsula, and my research site that I'm going to talk about is this huge mountain range that is bordering also the state of Kosovo and Shar Mountain. So I'm gonna go now to my presentation. Okay. So as you introduced me, John, thank you. My name is Maria. I'm a master's student at the University of St. Cyril and Methodius in Skopje. And I'm going to present you some results from my bachelor thesis, since my bachelor degree is in ecology. So while I was listening to this previous, especially the session with the wetland ambassadors yesterday, I couldn't ask myself how I got into wetlands and what brought me here to this conversation and to this conference, sorry. And it's interesting that I'm a new member to the Society of Wetland Society of Wetland Scientists. Just this year I joined the society, particularly the European chapter. So these are a few stepping points towards my goal in working research in wetlands. The plant flora is very rich. In Macedonia, it's recognized as one of biodiversity hotspots in Europe, especially in the Balkan as well, which includes a lot of endemic floral and fauna species. The problem is that there is really a significant gap of knowledge of wetlands in Macedonia. There are some partial phytocenological studies, but regarding their ecology, there is really a lack of knowledge. And on the other side, wetlands are considered as threatened habitats in the National Biodiversity Strategy and Action Plan. And according to the law, they are according to the law of nature conservation in Macedonia, they are considered as or designated as protected ecosystems. So why high mountain mires? Because my previous knowledge about wetlands in Macedonia is that the lowland wetlands were pretty much on large scale drowned out. And they previously occupied around 1% of the total country area. And because my main interest is how ecosystem functions, I wanted to take an ecosystem that is not anthropogenically disturbed. So that's why I switched to the mountain ecosystems. And stratigraphic, as we call them, or the mires, occur in Macedonia above 1,300 meters above sea level. And most representative sites with these ecosystems are on Shar Planina, Jakupica, Osugovski, and other high mountains in Macedonia. So that's how Shar Mountain came out. And I showed you the mountain range on the Google Earth image. So Shar Mountain is a huge mountain. Up to the 19th century, it was actually known by the name Catena Mundi, which on Latin refers to the chain of the world. It was considered that it's the highest mountain in the Balkans. It's very diverse. It has a lot of endemic and relic species, especially rich in flora, and it has a lot of peaks above 2,000 meters above sea level, which is really which can show the real influence of the alpine climate. So I picked few sampling size, sampling plots, especially mountain. Even though its geology is very complex, here in the central part is mainly limestone, which is especially important for some plant species. So what I was trying to learn, I was trying to do some initial research upon the ecology of this ecosystem, because as I said previously, this haven't been done at all. So there is no experience, and there is a lot of lack of knowledge in this field. Goals I set were floristic composition, plant diversity and biomass, plant chemical composition, including phosphorus and some biogenic nutrients, soil properties, including typification, water capacity, total carbon and nutrients, analysis of the main limiting factors, and some identification of future research goals. So I choose, as I said, 6 transects in the central part of the mountains that were distributed from about 1,800 and up to 200 meters above sea level. There are small plots, small ecosystems that are scattered, but represented with good number of them on this mountain. So in all 6 mires, we excavated soil profiles, and we analyzed basics. And according to the water capacity and volume density, we got some primary numbers, and this was also somehow known before, because some pedological research on similar soils had been done, and data was available in literature, so we could have compared. Regarding the plant diversity, we registered species of 47 taxa, and we managed to detect some Balkan endemic species, which gives a greater value to this ecosystem. It is important to be mentioned that we didn't do phytocenological analysis, but it is supposed from the literature that these mires belong to the phytocenological order of Carycetalia fusce, which is very very important and very rare in South Europe, not so common. Regarding the biomass, we use for the calculation the Shannon diversity index, and we calculate the diversity, which gave us information that these 6 plots have relatively low, are low productive. The mineral contact we also studied, because of we wanted to find out if we talk about minerotrophic group of mires, or are they geogenic. So also, one of the main points of this research was the relationship between the plant biomass and soil phosphorus contact in the top soil horizons, where we got some logarithmic correlation. So what from now, we did this basic research, but now I'm happy to share with you that we've started this year in support with a program from the Macedonian Ecological Society, a project that is also regarding the ecology of the forest mires, also on Shar Mountain. And I would like to point out, besides the previous research where I was doing it mostly alone with my mentor, now I have 5 young undergraduate students that will join me, and I'm hoping that we will enlarge this wetland community in Macedonia on a bigger scale. I'm hoping that you guys will help me with that, especially with networking and giving me advices about and share your experience with your work. So thank you for the opportunity, and I'm open for any questions you might have. Thank you.
Okay, so my name is Amanda, and I am a PhD candidate at the University of Toronto, and I work with Dr. Sarah Finkelstein. So today I will be talking very briefly about some of my dissertation on carbon dynamics in freshwater marshes. So wetlands are known for containing large carbon pools and for their potential to influence the climate. Most work to date on carbon pools in wetlands has focused on carbon pools in northern peatlands, such as bog and fen soils, as well as in coastal saline ecosystems. However, less work has been done on some temperate region wetlands in the northern hemisphere, and so carbon soil stocks are poorly constrained in some wetland types such as swamps and freshwater marshes. So my work is focusing on freshwater marshes in North America. And this is because they're one of the most prevalent wetland types and highly susceptible to alteration, particularly for agriculture and urban development. Freshwater marshes are not often regarded as carbon sinks and are fairly known for having minerogenic soils. A recent synthesis that I have conducted suggests, though, that freshwater marshes can contain organic rich layers in their sediments, so in their top sediments, but also in deeper sediments at these sites. As well, rates of carbon accumulation over long periods of time, so over hundreds to thousands of years, may be more significant in freshwater marshes than previously thought. However, these estimates, so these are soil characteristics, these estimates of soil measurements such as carbon contents and rates of carbon accumulation are highly uncertain due to a lack of field data. So in order to better constrain this carbon pool, more field data and work is needed. So some of my general research objectives are to better estimate carbon pools in freshwater marshes and their capacity to be net carbon sinks. And in terms of their capacity to be net carbon sinks, we're interested in understanding this over shorter periods of time, so over the last few decades, but over longer periods of time too, so over hundreds to thousands of years. As well, we seek to understand how internal mechanisms at these sites and environmental conditions affect carbon burial in freshwater marshes. Okay, so some work that we've been conducting more recently on this has taken place in southern Ontario, and one of our main study sites is Central Big Creek Marsh. And it is located in a coastal ecosystem on Lake Erie. And this system is influenced by the Big Creek River system. So from this site, we've taken a long sediment core that is more than 4 meters in depth. And we've taken this core back to our lab to conduct analysis using paleoecological techniques. So paleoecology is the study of past environmental conditions. And by using sediment archives, we can look at fossils and biogeochemistry to understand what's driving carbon burial over different time scales at these, in these freshwater marshes. Oh, thanks for the time reminder. So with this core that we have collected, we've been taking bulk density measurements and measurements of carbon contents to estimate the total soil carbon stock of this site. We also are employing dating techniques to estimate rates of carbon accumulation. So lead-210 techniques will be used to look at short-term rates of carbon accumulation in the soils of this site. So this is rates of carbon accumulation in the soils over the last few decades. And we've also been employing radiocarbon dating techniques. And this is used to look at rates of carbon accumulation in the soils over the past few hundreds to thousands of years. And this information will be used to understand what proportion of the carbon that enters the top of the soil gets buried in deeper soil over longer periods of time without being re-emitted back to the atmosphere. So we have some very preliminary results, which I'm going to briefly show you here. So as you can see here, these are 3 figures showing measurements from our long sediment cores. So this is showing rates of vertical accretion, organic carbon contents, and rates of organic carbon accumulation. Our dates that we have at the moment suggest that this site may be up to 6,000 years in age. So that means that over the last 6,000 years, this site has been accumulating sediments, and some of these sediments can have organic carbon that gets buried and sequestered at this site. But something I want to highlight here is you can see here that so this is showing the top sediments by the 0 to 100, and then we have our deeper sediments down to 300 to 400. But what's happening in the top sediments is not, can be quite different than what's happening in the deeper sediments here. So this shows us the importance of considering these deeper sediments in case we have or layers that are more rich in organic matter that we wouldn't know just by looking at the top sediments. And it also shows us the variation that can occur in rates of carbon accumulation over time and in the deeper profile. So some of our next steps will be getting more dates, particularly in the top core, so we can have higher resolution here to see what's been going on in more recent years, as well as doing elemental analyses to measure total and inorganic carbon. And this will help us better estimate the total carbon stock and rates of carbon accumulation. And overall, to help us address our objectives to understand freshwater marshes as net carbon sinks. So thank you for listening, and thank you for those who have organized this symposium, and you can send me an email or message me over Slack if you have any questions. Thanks.
My name is Megan. I'm at the University of Waterloo, and my master's project is looking at greenhouse gas fluxes on restored peatland seismic lines. You may recognize some of this because Taylor and I are actually field partners working on the same site. So seismic lines, again, are used for exploration for oil and gas. And there's currently over 345,000 linear kilometers of them running across Alberta peatlands. So when they create these lines, the trees and vegetation cover are removed, and any small scale topography is flattened to allow that seismic equipment to move through. Now, they originally thought that these would recover naturally, but as Taylor mentioned as well, some of these lines are approaching 60 years or more, and they're still just as evident. The trees are not recovering, and they are actually flipped more to sources of methane rather than carbon sinks and stores. So we're looking at ways of restoring these. So we use 3 mechanical treatments to recreate the small scale topography and restore the conditions for tree growth, which is mainly drier spots to grow, as well as to restore the carbon uptake cycle rather than emissions. So we have the rip and lift technique, which is using furrows to create those highs and lows. We have inline transfer, which takes a chunk of peat off of the line and places it back onto the line. And then the hummock transfer takes a chunk of peat off of the adjacent natural area and places it onto the line. And then those create those hummocks and hollows. We're comparing those to natural and untreated sections of the line as well. So our site actually divides up into 2 sub-sites. We have a rich fen and a poor fen. So each of the treatments are duplicated in both rich and poor fens, and then all of my collars are paired between the hummocks and hollows so that I can calculate the fluxes on both. And I was taking fluxes for CO2, so carbon dioxide, as well as methane production over the growing season last summer. So some preliminary results. So just a quick review of what this actually means. Gross ecosystem productivity is the total uptake of the system of CO2, and that's typically the plants taking up carbon. And if we subtract ecosystem respiration, which is the emission mostly from soil microbial communities into the atmosphere, then we are left with the net ecosystem exchange, which is the overall change and direction of the CO2 flux. So positive numbers in these cases means that it's being released into the atmosphere, and negative numbers means that it's being taken up and stored as carbon in the ground. So overall, we found that all three restoration treatments reduced the carbon or CO2 uptake of the lines in the first summer. As well, for CH4, similarly, all three of the treatments increase CH4 emissions. So CH4 is a one-way measure, and it's produced under waterlogged conditions. And you can see here that the mounding techniques had the highest amounts of H4 emissions. And in particular, there's this one huge jump in the hollows of the inline transfer of the rich fen. And we think that might be just because they're the wettest and deepest, and it's also a Carex-dominated area that was beginning to reestablish partway through the summer. So what comes out of this? We're not really sure. This is the first summer that this is, or the first time it's been tried, and last summer was the first summer of the project. So we need more time to see if some of those greenhouse gas emissions are reduced, and if it actually allows for trees to establish and grow, and if it will actually revert back to carbon sink instead. And it may mean that different or even no restoration is better for different types of peatlands. So yeah, with that, I just want to give a quick thanks to my supervisors, Dr. Maria Strack and Dr. Scott Davidson at the University of Waterloo, as well as our collaborators and all of the field partners, and lots of helping hands out in the site and in the lab over the winter so far.
So my name is Maggie O'Brien. I am a senior undergrad student at Loyola University Chicago, and this is my independent research that I've been working on for the past few years, looking at invasive cattail residence time and how that affects plant communities and below ground processes. So since we're all wetland scientists here, a lot of you are probably familiar with with cattail at least. Sorry, lost my presentation. Okay, so Typha X glauca is a hybrid of native Typha latifolia and non-native Typha angustifolia, and it becomes much more aggressive than either of its parent species. It can shade out other plants, and because of its efficient nutrient use, it quickly becomes a monoculture in Great Lakes coastal wetlands specifically. So studies such as this one, Mitchell, have found that there are increases in Typha density, increases in Typha biomass in 1 to 10 years since the initial invasion, which makes sense, becomes more dense. And then in 15 to 20 years, there are decreases in plant species richness and diversity, and changes in the soil occur after 35 years. So I wanted to look at how the seed bank might be affected in the first 35 or so years. Because the seed bank holds sort of that potential to restore the native plant community in a more passive way than replanting. So we would assume that if the extant plant community is diverse, then the seed bank is also going to be more diverse. We hypothesized that the longer that the Typha has been present, the less diverse the plant community will be, the less diverse the seed bank will be, the less likely it will be to recover naturally after removal of the invasive species. So not only are we looking at whether or not the diversity of the seed bank decreases, there are important implications for site selection for wetland restoration and management, depending on whether or not certain sites are going to be more ready to regenerate after biomass removal. So this is where my sampling location was. Here we have Michigan and Lake Michigan. It was located in the St. Mary's River on an island. This is the river connecting Lake Superior and Lake Huron. Here's an aerial photo of the site. So each of these red circles is a Typha clone because they reproduce asexually. Each stand could be a couple of genetic individuals, but hundreds and hundreds of stems. And each of these you can see, except for one of them, but these were identified using GIS, going through and identifying the extent of cattail at different periods in time. So here's an illustration that I made. You can see the darker circles are the oldest, and the circles get lighter as you go out. And I sampled within each of these age ranges. We sampled along a random transect with a vegetation survey and took soil cores. The soil was cold stratified to replicate overwintering, and soil was planted in pots that were then put in a growth chamber to simulate a Michigan summer with the light, humidity, and temperature. Water was kept level at the soil surface to mimic mudflat conditions. And I checked the pots for seedlings and identified all the seedlings. Some of them I had to grow in a greenhouse till I could identify them, and did some chemical analyses for the soil. So I identified over 900 seedlings, but only 9 species, with 3 genera aside from that couldn't be identified as species. There probably the lack of diversity is due to the difference in the amount of time certain seeds of different species can persist. So I'm going to skip that. So the interesting finding is that there is not a significant difference in seedling diversity or in the total number of seedlings with time since invasion up to 25 or so years. There is a slight, there's a marginal correlation, marginally significant correlation between seedling diversity and vegetation diversity that we found, which makes sense. And this is interesting. If anyone's interested in this, please let me know. But as the concentration of soil nutrients increased, the total number of seedlings decreased, which may be because of TIFA's mechanism of invasion is it increases the nutrients by creating an excess of litter. So what we found is that sites that have been invaded for longer might still have good potential to be restored, similar to newly invaded sites. They might have that same regenerative capacity. And I would like to thank my mentors, Shane Lishua and Drew Monks, and my funding sources. And thank you.
Well, thank you everybody for being here today. My name is Chelsea Duvall. I'm a PhD candidate in soil science at the University of Wyoming. And today I'm going to talk to you about a portion of my dissertation research entitled Iron Monosulfide Concentrations Indicate the Presence of Hydric Soils, a case study on riparian soils in western Nebraska. Okay, so most people on this call, I'm sure, are familiar with the 3-parameter approach, which we use to identify wetland areas. This includes conformation of hydrophytic vegetation, wetland hydrology, as well as hydric soils or wetland soils. But one thing to note is what happens when one of these parameters does not meet the requirements for this equation. Meaning, when we have soils that meet the definition of a hydric soil, but they're not exhibiting any soil morphologies used within the field or used as a field indicator. This presents us with the situation of a problematic soil. This became a case on the greater North Platte floodplain located in Nebraska, where the NRCS back in 2016 identified these 4 sites just east of the Wyoming area, in Scottsbluff and Sidney, Nebraska. None of these soils met any field indicators of hydric soils outlined in the National Technical Committee for Hydric Soils Guide, as you see here to the left. Essentially, what these soils were lacking was the accumulation of organic matter or distinct redoxomorphic features, meaning iron concentrations or iron depletions, that we typically use to identify hydric soils in the field. One thing I should note is that these sites are located here on the Niobrara Shale Formation, meaning they have a significant sulfur source. Another thing that the practitioners noticed while in the field were these dark black splotches within the soils. It was not organic matter. It wasn't manganese oxides. So this is where we came in to identify what was going on in these soils. Coming from a coastal background and looking into the geology of this area, we started to question if these were iron monosulfide concentrations. For those who are not familiar with iron monosulfide concentrations, they are black insoluble soil morphological features that form exclusively under anaerobic conditions and further down on the redox ladder under strongly reducing conditions, where sulfate is reduced to sulfide. In order for these to form in the soil, you need anaerobic conditions present, sulfate-reducing bacteria, a carbon source for those microbial communities, as well as an iron and sulfur source, resulting in what you see here, which are these black formations. Very distinct morphologies. However, in these freshwater or salt-affected systems that are more inland, they're less common. So this led us to question, can the identification of iron monosulfide concentrations provide sufficient evidence to indicate reducing conditions in soils that fail to meet any other criteria for hydric soils? And this is where I should mention, within that white field guidebook, there is no mention of iron monosulfides in any form of the vocabulary or any indicators as it currently stands. So in order to conduct this study, we went to these 4 field sites to confirm the presence of wetland vegetation as well as wetland hydrology to make sure those 2 parameters were set in place. But we dove a little deeper into the actual morphology of the soil to confirm the presence of iron monosulfide concentrations, specifically using a 3% hydrogen peroxide solution, which induces a color change via rapid oxidation, where the black color increases in value more towards a lighter gray or brown. It's a very distinct color change, easy, very affordable to assess in the field. We also deployed indicator of reduction in soil tubes or films, and this was to confirm and assess reducing soil conditions, looking for those stronger reducing conditions. The standard for the hydric soils technical standard states that these tubes need to be deployed 30 days in situ. The orange iron oxide coating on that tube over time, if sufficient reducing conditions are present, that orange will change. It changes to a white color, leaving behind the white color of the tube if iron is reduced. However, also on this tube, you can get black iron monosulfides that precipitate if stronger reducing conditions are present. So what we found here, they look like fairly bland soils, but we actually went in and were able to confirm 1 upwards to 7% of iron monosulfides within the soil morphology, and that's in the upper 30 centimeters. We didn't recognize it at one of the sites, but that's okay. Looking into our IRIS results, just so everybody gets a picture of what these tubes look like in a drier environment. When they were deployed in the wetland system, we start to see this black color change. And noted here on the right, we're looking at 5 replicate tubes per site, and per recommendation of that Vaughn et al. 2016 paper, each of the 5 tubes met that at least 20% precipitation of FES. Leaning more towards at least 3 out of the 5, closer to a 10% color change on that tube. So we do have stronger reducing conditions. We have to question why they weren't all meeting the FE reduction requirement, but that's leading into future research. For sake of time, I'll just note that these soils did display evidence of sulfate reduction via the morphological evidence of FES, as well as FES on those IRIS tubes. And this will lead to our next steps, running additional statistical analyses to assess those relationships and using these data and results to propose the incorporation of FES into the field indicator of hydric soils guide. With that, I'll thank you and leave my contact information. I'm happy to talk through Slack or through the chat.
Hello everyone, my name is Carla Ayala and today I'm going to talk about mercury contamination in soils and biomass of a St. Lawrence River wetland. When the Moses Saunders Dam started operating, water level regulations began in the river. The disruption of the river's natural water levels had negative impacts on the ecology of wetlands upstream of the dam. Before the regulation, cyclical high water levels eliminated dense canopy emergent plants, and low water levels allowed less competitive understory plants to grow. This plant biodiversity supported fish populations. Maintaining the water level stable favored the expansion of the flood-tolerant taifa. This affected fish populations by decreasing the submerged aquatic vegetation habitat. Taifa's extensive system allowed them to accumulate sediments. Plan 14, enacted in 2017, is an ecological restoration plan that seeks to provide more natural water level fluctuations to support Plan 2014, enacted in 2017, is an ecological restoration plan that seeks to provide more natural water level fluctuations to support the diversity of this area without interrupting the dam services. This restoration plan has the potential to benefit the biodiversity of the wetlands, but high water levels may also release legacy mercury that has been accumulated in the soil and cactus biomass. Exposure to mercury represents a public health concern. It is a potent neurotoxin that affects cognitive abilities, behavior, and reproduction in mammals. The introduction of mercury into the river could cause mercury biomagnification. Our objectives are to quantify the mercury pollution in the wetlands and evaluate mercury pollution trends in other wetlands within the river. This is vital for evaluating the potential adverse effects that may have arisen with the implementation of Plan 2014. Our hypotheses are that mercury concentration in taifa organs reflect that in soil, and that mercury distribution in soils and biomass in a wetland are homogeneous. Our study site is a model wetland located in a state park in Wellington, New York. It was chosen as a model because of its accessibility. Its area may also be representative of the areas of other wetlands within the river. We took samples in 9 sampling points. From each sampling point, we took 4 cactus individuals, 1 quart Ziploc bag full of dry leaves, and 4 soil samples. We had in total 81 samples to process. We processed the samples in the lab. Plants were cleaned and divided into 4 organs: leaves, root core, stem, and roots. After processing, all samples were run in the mercury analyzer and we measured total mercury. The Kruskal-Wallis test was used to determine if the mean mercury concentrations of sample materials differ significantly. Correlation analysis was used to determine if mercury concentration in cactus organs is a good indicator of mercury concentration in soil. To evaluate the potential for bioaccumulation, we calculated the bioconcentration factor, or BCF. The BCF is useful for determining how efficient taifa are in taking up mercury from the soil and incorporating it to its tissues. In the graph, we can see the average mercury concentrations of different sampling materials. Roots had the highest mercury concentration, while stems had the lowest. We can see that there is no significant difference in mercury concentration of roots and soil. No significant correlations between mercury concentration of soil and mercury concentration in cactus organs were found. The average BCF was 1.64. Our data do not support our hypothesis. There are significant differences among the mercury concentration of sampling materials, and no significant correlations were found. This contrasts with other research in which significant correlations between mercury concentration of soil and roots were found. However, most of these studies are carried out at sites with high heavy metal contamination. Our study site is not as heavily contaminated as the sites in other studies. A VCF higher than 1 may indicate that the plant functions as a hyperaccumulator of mercury. Our study contributes to the understanding of mercury contamination in the river's wetlands, which is limited. If further studies demonstrate significant potential of mercury biomagnification, Plan 2014 should be re-evaluated to ensure the health of humans and wildlife. I would like to thank my mentors, Professor Michael Twist, for his guidance and for considering me for this opportunity, and Evie Bromstead for teaching me about wetland science with the utmost patience and kindness.